medicine is a very important medicine, and the basic immunology behind it, we will describe it today. So in my last class, we have described the MHC1. You can remember the major histocompatibility complex and human leukocytic antigen. The human leukocytic antigen and MHC are the same because the major histocompatibility complex, these are the class of gene. Those gene MHC1 and MHC2, you know, those are presented outside when any antigen received by the body. Say for example, in our cell, the exogenous and endogenous antigens are processed in two different ways. In my last class, I described it because the exogenous antigen, they are presented by MHC2 and the endogenous antigen are presented by MHC1. So the MHC1, the major histocompatibility complex, these are a sequence of gene by which a receptor-like material, a receptor-like projection is displayed by the nucleated cells and macrophage cell outside. In case of any exogenous antigen, they extend the MHC2 molecule by which, sensing the MHC2, our surveillance cell, immune cells, can be able to recognize what type of immune response that particular cell is going to elicit. Similarly, in case of MHC1 presentation of endogenous antigen, viral antigen, graft antigen, these antigen are processed endogenously and are expressed by MHC1, and directly it is going towards the cytolysis, that is by TC cells. So there is a possibility of presentation of these MHC1 cells to T cells, and T cell name break down to its subset like TC cell, Th1 cell, Th2 cell. Last day I have explained this Th1, Th2 response. And it can also happen that so many clonal proliferations of different type of T cells are there. TC cell, T cytotoxic cell, is similar to CD8 cell, and as well as TDTH cell. The delayed hypersensitivity reactions is displayed by these TDTH cells. They can display it to TDTH cells. So immune stimulations, Th clonal proliferations by MHC2 may respond in different way, Th0 to Th1, Th2. Interleukin secreted from Th0 can stimulate TC cell, that is CD8 cell, and CD8 cell has cytolysis capacity, it can lyse the cells. Similarly, Th1 can stimulate cell-mediated immunity. Th2 can mediate humoral antibody by liberating 2, 3, 4, 5 interleukins and ADCC response, and can kill the cell by liberating different enzymes. So this pathway is clear to you now, Th pathway, Th subset. Another type of subset is there, that is TDTH cell, that is responsible for delayed hypersensitivity, and as well as it can stimulate those TC cell. So it is a different route by which it can stimulate TC cells. Now if we see, if we analyze the MHC1 and MHC2, these MHC1 and MHC2, these are the class of genes, and they are present in the chromosome, and especially in long stretch. First, it is experimentally proved in mice. That is why in mice cell, it is displayed as H2 cells, and from that H, and as well as from human leukocytic antigen, these are seen in human leukocytes. It is known as H. The H is human. And human leukocytic antigen, as it is displayed on the surface of the leukocyte, all the leukocytes. So these are leukocytic antigen. Now their gene are typically seen in three particular area in our system, in our chromosome. Now, the particular three loci or locus. One, the gene arrangement is like this: A, C and B. In case of 1, HLA 1, the system is arranged like this: A, C, B. Similarly, the gene aggregation in case of MHC2 is DP, DR or DQ or DP, DQ, DR. This is the system. This is D locus. So A, B, C locus and D locus, these ABCD locus are important for us for displaying MHC1 and MHC2. The genes are there within our system, and genes are displayed through their genomic expression by protein, by the cells to outside. That MHC2, MHC1 is displayed and maintained by the gene inside our cell. So, HLA3 gene complex is mostly related to complement. They are the complement activating gene. Their protein is complement. So, the ACB responsible for MHC1 display and DP, DR, DQ responsible for MHC2 display. Now, how the structure is that? What about the structure? MHC1 structure, if you see, it is the cell. Outside the cell it is displayed, MHC1, and the cytoplasmic tail is there, anchored in the cytoplasm, and outside the cytoplasm, alpha chain and beta chain are there. The alpha chain is alpha 1, 2, 3, 3 domain are there, like immunoglobulin domain, and beta 2 microglobulin is attached with this alpha chain. So this is the complete chain where only one anchoring cytoplasmic tail is there in case of MHC1. But in case of MHC2, 2 cytoplasmic tail is there, one for alpha chain and one for beta chain. One alpha 1 and alpha 2, and otherwise beta 1 and beta 2. Now this particular domain, the groove, this is called groove. This is the groove which is important, where it represents the particular type of antigen. When this MHC1, that means within the cell, this is the cell, within the cell, any endogenous antigen is processed and they are displayed by this particular MHC1 molecule. They are processed and displayed. In case of MHC2, this is our cell. Any exogenous antigen, they are processed and displayed by the domain, by this group. This is MHC2. The molecule looks like that. This is the molecule of MHC1 and this is MHC2. By sensing the genomic structure of this, our CD4 cell and CD8 cell, they start eliciting response. CD4 cell, they can sense MHC2. CD8 cell can sense MHC1. Not only the antigen is the main thing. The main thing is sensing this receptor. Now, the entire response is elicited. Is it clear? This is all about these two, MHC1 and MHC2. Now, the different type of immune response are seen in case of transplant antigen. It is very difficult to read from book. The basic thing is that all the reactions, immune response in transplant medicine, try to analyze them on the basis of T cell response and B cell response. The graft is something, maybe tissue, maybe organ. It is placed on the recipient cell, a different site, and the graft, when it is from another patient, having this different type of the genomic sequence. This HLA complex is multi-allelic. That means A, C, B contains at least 54 alleles. That means variation of gene. Gene variation is called allele. So 54 types of variation is possible in B. Similarly in C, 11 type of variations is possible. And A, 24 type of variation is possible. Now one gene is coming from father, another is coming from mother. So always there is two type of expressions, one from mother and another from father, from parents. And here the two expressions is ever changing. Different type of alleles are there. Say for in case of B, the 10 domain is expressed, and in A, the 1 domain is expressed. So that character will be 1 by 10. So different type of permutation combination is possible in human being. And each and every patient, each and every person, by their fingerprint, is different from other. By these location, by this HLA complex. So, any transplant from one person to other person, there is mismatching, the possibility of mismatching of these complexes, of these genes. It is really threatening. Different type of graft is possible. Autograft, from my own body, one particular cell transplanted to other cell, other region. This is autograft from the same person. So, no tension of mismatching. No tension of mismatching from same person, the same graft to another region. Autograft. Acceptability is 100%. 100% acceptability. In case of isograft, the identical twins, the HLA complex is same, the acceptability is 100%. But in case of allograft, from one human being to other human being, from sheep to sheep, tiger to tiger, acceptability is variable. Chance of rejection is more. And here lies the real challenge in life. Because we are not getting always autograft, we are not getting isograft always. So we are going for allograft. So we have to face the challenges. How to accept it. How to tackle the immune response. How to manage it. This is the transplant medicine. Xenograft concerned with this, allograft. So one person to another person with different HLA sequence. Two types of reactions occur. The first set of reaction and the second set of reaction. The graft is there. Initially, there is no vascularization at the graft site, no blood supply. Initial 3 to 4 days are uneventful, beautiful. Patient is happy, family members are happy. But after 5th day onwards, the neovascularization start. That means new blood vessel development start from the recipient's body towards the graft. And with this neovascularization, the surveillance cells, immune cells, T cells, B cells, they are there. They are now entering the graft. What is that? It is not matching. The graft started presenting their immune cell, their existing cells to the system by MSC1 or MSC2. Recipient cell receiving this, clonal proliferation of T4 cells, TC cells activated, plenty of cellular invasion starts. Suddenly, the vascularization is cut off. No vascularization now. No blood supply now. Cut off. Something new. Rejected. TC cell started secreting granzymes, different enzymes over there. It is something outside from our body. Rejected. No vascularization, no blood supply. There is ischemic necrosis and the graft is rejected out. So this is the first set of response. The second set of response, our surgeon, initially what we thought, oh my God, something is there. There may be infection. There may be something. Go for another graft. So from the same patient, from the same donor, another graft is taken and it is implanted to the same recipient. At that time, immune response was not clear to us. But what happens? The immune cells, those are primed. Those are sensitized immune cells, memory cells. From the initial primary response, the secondary response. Just think about the immune response. Primary response and secondary response. Persisting antibody is there. T cells are primed. They are suddenly stimulated. Once again, the same enemy. They don't know that what is graft. The same enemy. Kill it. There's an emergency. Quick vascularization and quick entry of the cells. Initially, there is 4 to 5 days lag. Now there is no lacking. Immediate action from the second set. Violent reaction. And within 1 week, the graft is rejected. That is the second set of response. The immune response is these memory cells are the main important cells. Those are stimulated. So we have the different route now. Either we have to suppress the immune response of the recipient, or we have to bypass that particular graft. We have to kill all the immune cells of the graft. There shall be no response. There will be no cells. Or by different system, by different drugs, we have to bypass the immune response. And we have to give time for the adjustment. So that is the key of the success of the response till date. The response is 70%. There is 30% rejection is still happening. No 100% success in case of allograft. So this is all about the transplant medicine in a nutshell. This thing only you have to remember this much. Other subject beyond this is the scope of transplant medicine and transplant surgery. They will tell the acceptability criteria, how to make them success. There are different ways now. So this much for this class. একদম আমি জিস্ট করে ক্ষীর করে এটাকে তোমাদের প্রেজেন্ট করলাম। এবার তোমরা এখানে স্লাইড দেখবে, গোলমেলে হয়ে যাবে ব্যাপারটা। স্লাইড যত দেখবে মনে রেখো এটা হচ্ছে সারমর্ম। যেটাই ঘটুক সেই ইমিউন রেসপন্সে নিয়ে ফেলবে, সেই টি সেল, সেই বি সেলস, টি সেলের কাছ থেকে ক্লোরাল পলিফারেশনস হবে, টি সাইটোটক্সিক সেল স্টিমুলেটেড হবে, অথবা ডাইরেক্টলি টি সেলকে ওরা প্রেজেন্ট করতে পারে গ্রাফট নিজে। আরেকটা রিয়াকশনস আছে যেটা গ্রাফট ভার্সাস হোস্ট। এখানে হোস্ট ভার্সাস গ্রাফট হলো, যেখানে হোস্টের রেসিপিয়েন্ট সেল সেই ইমিউন রেসপন্স দেখালো গ্রাফটের এগেনস্টে। সিমিলারলি উল্টোটাও হতে পারে, গ্রাফট ভার্সাস হোস্ট, যেখানে গ্রাফটের মধ্যে ইমিউন সেলস আছে, অলরেডি এক্সিস্টিং সেলস, এবং রিসিপিয়েন্টের মধ্যে নেই, সেখানে দ্য সেম রেসপন্স উল্টো দিকে হবে, দ্য আলটিমেট ইজ দ্য সেম, গ্রাফটটা রিজেক্টেড হয়ে যাবে। এখন আই হ্যাভ ওয়ান কোশ্চেন, সেটা হচ্ছে fetus and mother relation. Is fetus a graft? Fetus কি graft? Fetus কেন রিজেক্টেড হয় না? Fetus কেন রিজেক্টেড হয় না? এই প্রশ্ন রইল তোমাদের জন্য, এই প্রশ্ন রইল তোমাদের জন্য। আমরা পরের ক্লাসে এটা আলোচনা করব। হ্যাঁ, এটা তোমরা ভাবো, পড়ো, আমার একটা প্রশ্ন দেওয়া রইল যে ফিটাস কেন অ্যাবর্টেড হয় না। চলো এবার স্লাইডগুলো দেখা যাক। তাহলে মেজর হিস্টোকম্প্যাটিবিলিটি কমপ্লেক্স, the complex is a group of genes, তোমরা দেখতে পেয়েছো ছবি, সেই গ্রুপ অফ জিনস কোডিং ফর এ সেট অফ হোস্ট সেল সারফেস মলিকুলস দ্যাট বাইন্ড টু পেপটাইড ফ্র্যাগমেন্টস derived from pathogen, endogenous, exogenous, display them on host cell surface for recognition by the appropriate T cells, displayed on the surface for appropriate T cells activity. Present in almost all the human cells, but first discovered on the surface of leukocyte. Hence, in human, the MHC-coded proteins are also called as human leukocyte antigen or HLA, serve as a unique identification marker for every individual. Following transplantation of a graft, the recipient mounts an immune response against the graft's MHC molecule and vice versa. The acceptance or rejections of the graft is directly dependent on the MHC molecules of the graft and the recipient. As the MHC molecules determine the compatibility between the graft and host tissues, they are named as histocompatibility antigen. The MHC gene and products located in short arm of chromosome, এটা কোথায় থাকে? এটা থাকে short arm of chromosome, short arm of chromosome থাকে। HLA complex extends 4000 kbp length, covering more than 1000 genes, and genes are clustered in 3 regions named as MHC region 1, 2, 3, and these are multi-allelic. Each genes are multi-allelic. This is the picture. More easier form for examination purpose is this. The major histocompatibility protein class MHC1 and MHC2. The cytoplasmic tail here, it is 1, and this is the structure of MHC molecule, class 1 and class 2. B2 or beta 2 microglobulin with a chain is necessary for the expression of MHC1 molecule on the cell surface. Cell surface expression is required. Antigen peptide group, peptide group. For CD8 cell and for CD4 cell. This is the difference between class 1 MHC and class 2 MHC for theory question purpose. Present on all nucleated cell except sperm, and MHC2 antigen presenting cells only. Peptide antigen is the CD8 and CD4, endogenous and exogenous. The peptide antigen of the processed one is 8 to 10 and here is 13 to 18 amino acid long. This pathway is known as MHC class 1 pathway is known as cytosolic pathway, and MHC2 is endocytic pathway. Gene expression regulation is not for you. The different type of gene defect may produce different type of disease. Defect in these particular chains may result in HLA-B27, multiple sclerosis, myasthenia gravis, rheumatoid arthritis, the collagen group of disease. Now, the transfer of graft. Transfer of graft or transplant, that is cells, tissues, organ from one side to another is called graft or transplant. The individual from whom the transplant is taken is referred to as donor, and individual to whom it is transplanted is known as recipient. The classifications, most important classification is autograft, isograft, and allograft and xenograft, and histocompatibility matching, the graft and recipient, the different factors to consider. Most importantly, MSC molecule and ABO-RH compatibility. The first, for the acceptability of graft, we go for the blood grouping of the donor and recipient. So first is blood grouping is required, followed by MSC molecule matching, the MHM molecule matching. The immune response is already known to you now. The graft rejections may be acute within one week period, may be chronic when TDTH pathway is taken, and hyperacute within days, 4 to 5 days it is rejected out. T cells and B cells, acute graft rejections. Just remember first set and second set of reactions. This is the comparison of different type of graft rejection, hyperacute in minutes to hours, and acute in weeks to months, and chronic in months to years. This one is mediated by TDTH cell, that is type 4 hypersensitivity reactions, and acute by cytotoxic T cells, chronic DTH cells, and is the preformed antibodies, anti-ABO and anti-HLA is the responsible for hyperacute case. Different factors and tissue matching is now important. This is the acceptance criteria. Initially neovascularization, initial blood vessels formations from recipient towards the graft. Within days 3 to 7 revascularization takes place, and days 7 to 10 the healing, and days 12 to 14 resolution. And type of primary rejections when an allograft is placed for the first time from a donor to a recipient. And type of primary rejection when an allograft is placed on the first time from a donor to a recipient. The first set of response, vascularization, and then with vascularization from 7 to 10 days cellular infiltrations, and now the tissue necrosis and thrombosis starts. The second set, quick vascularization followed by the cellular infiltration within day 3 to 4, and day 5 to 6 there is thrombosis or necrosis and rejection. The mechanism involved in graft rejections. The donor MHC carrying allogenic MHC antigen. This is the MHC antigen, the donor MHC. Direct presentation to CD4 cell. And with the presentation, immediately there is IL-2 formation, and IL-2 stimulating T cytotoxic cell may stimulate IL-24, IL-5-6 by B cell stimulations, TC cell stimulation, and TDTH cell stimulations. So in case of graft rejections, 3 subsets, these are the important sets. These to remember. If you remember this picture, the immunology of graft will be very clear to you. So clonal proliferation of 3 sets, TC cells, B cells, and TDTH cells via TH cells. First is stimulate TH cells. TH cell stimulation, liberation of interleukin 2, liberations of interleukin 2, 4, 5 and 6. It will go for B cell, and IL-2 stimulate T cytotoxic cell. And T cytotoxic cell, CD8 cell is the same thing. Through MHC1 receptor expressed antigen and membrane damage, it will secrete some cytokine as well as some granular material, toxic material, and destroy the graft. The ADCC response is antibody dependent cytotoxic response via B cell, and the same thing, the killing of the graft. And here the TDTH, the delayed response, and where interferon gamma and tumor necrosis factor B is responsible, and similarly graft is rejected out. So three different pathway. The result is same. The cells involved are effector cells, B cells or T cells, whatever it may be. How to prevent? So matching of performing blood group for the recipient as well as the donor. Recipients we know from the donor, and as well as HLA typing. And now the PCR methods are also there, but the confirmatory method what we perform is microcytotoxicity or mixed lymphocyte reactions test, usually done for tissue matching. This is the simple picture to remember. The lymphocytes are HLA typed by cross-matching to panel reactive antibodies using the complement and dependent cytotoxicity. The CDC test is called. The Buffy coat from the patient, that is from the donor, is taken, and the Buffy coat, the monoclonal antibody, is tagged with this microwell ELISA. This is an ELISA plate where the monoclonal antibody is tagged with the different expected antibody. These antibody what we expect here, the different types of antibody are there, and they are tagged with these microwells. And the positive reactions to antibody kill cells, then when the antigens are given, when antigens are supplied, this is an antigen supply from outside. This is the patient sample is given when the patient samples that contains the antigen. Antigen antibody reaction will take place with the monoclonal antibody, and then we add complement to lyse the cells. The lyse cells are then stained with trypan blue. The lyse cells are stained by trypan blue, and we will be able to identify the concentrations of lyse cells. With the concentration of lyse cells, we will be able to understand how much concentrations of the antigens are present on the tissue cells, on the recipient, the donor cells. The negative reaction is always acceptable. No reactions, so it is acceptable. So this is microcytotoxicity cell, just to show you the microcytotoxicity. Immunosuppressive therapy, that is how to make the graft acceptable. Different immunosuppressive drugs are there, like steroid, and other than steroid is methotrexate and cyclosporine we can add to control the immune response of the patient, of the recipient. The corticosteroid, cyclosporine, and tacrolimus is another molecule now, very recent molecule. And these are the different procedure, newer generations molecules. These are immunosuppressant drug used. This is all about the transplant. Transplant লিখতে গিয়ে যা কিছু হয় গিয়ে ইমিউন রেসপন্সে ফেলে দেবে। শুধু তিনটে সেল মনে রেখো যে ইমিউন সেলসগুলো যখন টি সেল স্টিমুলেটেড হবে তখন তিনভাবে স্টিমুলেটেড হবে। একটা ডাইরেক্টলি টিএইচ ক্লোনকে স্টিমুলেট করতে পারে, একটা বি সেলকে স্টিমুলেট করতে পারে, একটা টিডিটিএইচ সাবসেটকে স্টিমুলেট করতে পারে। টিডিটিএইচকে করলে ডিলেড হবে, আর বাকিগুলোকে যদি করে তাহলে অ্যাকিউট রিজেকশন হবে। আর যদি সাবসেট যদি মনে রাখো যে পরে সেকেন্ড ইমপ্লান্টে কী হতে পারে, আজকাল আর সেকেন্ড ইমপ্লান্ট হয় না, তাও সেকেন্ড সাবসেট হতে যদি সেকেন্ড টাইম আবার ইমপ্লান্ট হয় তাহলে কিন্তু ওই একই পেশেন্টের, ওই হতভাগ্য পেশেন্টের ওই পরিণতি হবে। এখন কিডনি ট্রান্সপ্লান্টের অ্যাকসেপ্টেবিলিটি খুব বেড়ে গেছে আজকাল, প্রায় 15 থেকে 20 বছর পর্যন্ত ভালো যদি ট্রান্সপ্লান্ট হয় তাহলে থাকে। আরও চেষ্টা হচ্ছে বোন ম্যারো, লাং হার্ট, সিঙ্গাপুরে তো হয়েছে হার্ট সার্জারি, খারাপ হয়নি, ভালোই হয়েছে। বোন ম্যারো তো আগেই হতো, বোন ম্যারো ফেলিওরটা বড্ড বেশি বোন ম্যারোতে, তাহলেও হয়।